Next, we have the opportunity of meeting Dr. Badri Rengarajan. And remember to please review the bios in your booklet. He is the medical director in solid tumors at Jazz Pharmaceuticals. Let's give him a round welcome.
Thanks, everyone. It's a real honor to be here. So I joined Jazz about 4 years ago to help launch Lurbinectedin, which is our drug for small cell lung cancer, but also to launch the company into the solid tumor space, which was new for us. And while my work as a medical director is definitely useful here for this presentation, actually the thing that I think in my experience that is very relevant here and really gives me empathy for everyone is that I've, for several years now, been in the leadership of a patient advocacy organization that helps patients with 2 autoimmune skin diseases. These are ultra-orphan, and it was funny, my mom had one of the diseases, and this was at a time when the internet was not well developed. There was no Google yet. So when Google finally came around, or some internet search engine, I looked around and said, oh, there's this patient organization that's here to help patients. Raised my hand, volunteered, and I said, I'll do anything. You want me to bring coffee, make copies, whatever you want. But then I must have said something competent or done something competent. They actually invited me to be on the board. So I've been on the board for a long time. But the reason I say that is just to tell you that, a couple things. When I joined, I said, here we are in an ultra-orphan disease. How much can we really do? But what I learned very quickly in talking with patients, working with patients, is that we can do a lot. Small things go a long way, whether it's helping a patient find a doctor, whether it's helping doctors educate themselves on the landscape, whether it's improving the patient's quality of life, really small things, but really big impact. And I feel the same with you guys listening to your stories. So I say that simply to tell you that I get you and support and applaud the courage and the commitment you have to learn and to help each other. It's really fantastic. So with that, I'd like to introduce you today to Jazz Pharma and our activities and aspirations in cancer, and particularly lung cancer. So what I want to do is give you a little bit overview of our company, talk a little bit about our pipeline, and then in particular focus a bit on Lurbinectedin, or Zepzelca, which is our drug for small cell lung cancer. So let's start with Jazz itself. So Jazz was founded about 20 years ago, and our purpose is very simple: using innovation to transform the lives of patients. It's a company that was founded to help patients with rare disease, high unmet need diseases. You're not going to see us in hypertension or diabetes. It's a part of the fabric of the company. It's a part of the DNA of everyone who works there. Let's take the hardest diseases. Let's take the ones where patients don't have options, and let's see if we can make things better for them. Like I said, Jazz has been around for 20 years, and so we've become kind of a global company in that time. And as you can see from the map here, the yellow are places where we have direct presence and get our medications to institutions that help patients. The red is where we work with partners, and the white, still to be determined. But as time goes on, I hope more of this map becomes red. I hope more of this map becomes yellow as part of our global domination strategy. And I'm just kidding about that. Really, it's to help patients wherever they are, and we're very committed to that. So we've got kind of a portfolio of drugs that really hit 2 therapeutic areas. One is neuroscience on the left, and then oncology on the right. And on the left, we have some drugs for epilepsy, for sleep disorders. And on the right, we have Defitelio for veno-occlusive disease, Vyxeos for acute myelogenous leukemia, Raleigh for acute lymphoblastic leukemia, so some hematologic malignancy. And then, of course, relevant to this audience is Lurbinectedin, or Zepzelca, for small cell lung cancer. And let me just take a step back. You might ask, why in the world are we called Jazz Pharmaceuticals? That's kind of an interesting name. But one of our founders, who's our current CEO, Bruce Kozad, is a musician. And when he pulled this company together with his co-founders, they said, let's do something different, something new. Really focus on patients, really focus on having the employees have a good experience here. And then let's take these principles that a jazz ensemble or jazz band would have, right? Structured improvisation, collaboration, and personal excellence. And let's bring that to a pharmaceutical company. So structured improvisation, we're a very regulated industry. We have a lot of checklists, things we have to do. But within that, can we innovate and try new things? Collaboration, working together just like members of a jazz band. And then personal excellence. Let's really excel in our particular role and pull this all together to help patients. I'm not going to go through this entire timeline, but like I said, Jazz was founded about 20 years ago, and it grew by acquisition of other companies, by acquisition of different drugs, and it's just steadily grown since then. But of relevance to us is, let me see, The part in red, which is lurbinectedin, which was approved in 2020 for second line and beyond small cell lung cancer. And then since then, we've acquired a company called Jazz Pharmaceuticals, which has a cannabinoid platform and some other drugs along the way. Okay, just a quick word about our pipeline. I'm not going to read all of this. There's a lot of drugs, a lot of trials here, but that's not what I want you to take away from this. What I want to focus on are the colors. I want to focus on the number of columns and the height or the depth of each column to illustrate where Jazz has come from. So if we were talking about 15 or 20 years ago, the drugs we would have would pretty much be in the last column, your phase 3 and things that are commercial and marketed. But over time, what has happened is the company has grown an R&D infrastructure. So it's not only got marketed drugs, it's got phase 3 drugs, and it's going all the way back, phase 1, phase 2, and preclinical, trying to bring things from outside, develop them within, or develop molecules from within. So it's really the company has transformed quite a bit. The other thing is, if you look at the blue and the red, this is kind of interesting. So again, if we were talking 5, 6, 7 years ago, we would be in the red zone, which is neuroscience, really focused on sleep and sort of neuroscience types of diseases. But in the last, I would say 5, 6 years, the company has made a very big commitment to oncology, and those blue regions have grown significantly. The company is really doubling down and trying to develop that further. So a real commitment to oncology, and like I said, we're already playing in small cell lung cancer. And interestingly, if you look at the blocks with the yellow dots, that represents new things that have only come to fruition, have been initiated in the last 5 years. So really growing rapidly and really committing, again, to that blue zone. And a couple of compounds, I would say, beyond small cell lung cancer. We have we in-licensed a compound called zanidatamab from a company called Zymeworks. Really interesting geometry in that compound. It targets HER2 expressing cancers, and many of you may be familiar with HER2 from breast cancer. And we'll see a lot of trials starting up in that, and we'll see where it goes in terms of what cancers it ultimately gets applied for. Okay, so let me switch over to lurbinectedin now. But first, a kind of a story. So imagine you're in Spain, in coastal Spain, and there's this man who is a sort of very successful industrialist. He's very successful in the fisheries industry. And one day, this is years ago, and he's walking with his 2 sons, and he asks them, like, what do you want to do when you grow up? Like, what do you want to do with your life? And the first son says, oh, well, I want to learn business, I want to learn fisheries, and I want to be just like you. I want to be a very successful industrialist in the fisheries industry. Well, gold star, the father is beaming, says, that's the right answer, this is terrific. The second son says, well, I want to go out and get a PhD in biochemistry, and then I want to go into the ocean and see if we have interesting compounds we can find. Not a good answer, total loser. But it turns out he went on to get his PhD in biochemistry and ended up pursuing this passion for finding things under the sea and started a company called Pharma, and then Mar, which is Spanish for sea. And that's an interesting approach that the company's got. And that's kind of the origin story for actually lurbinectedin. So they found, they do some very interesting things. I'll say the founder observed, when you look at marine invertebrates or like urchins in the ocean, they don't seem to have a lot of defense mechanisms. They don't have big claws, they don't shoot darts at you, they don't do any of that, but they survive. How do they survive from predators, and how do they don't get cancer? What's going on? So he hypothesized that there's some compound, some chemical, hormone, whatever in these organisms that is protecting them somehow. But if we could go out there and isolate these things, it might give us ideas or actual substances to use medicinally. And it's not unlike if you look at pharmaceutical development in the 20th century and even before that, when people would go find these animals and extract some rare hormone or something from them, or go into the Amazon rainforest and find some exotic plants or animals and try to extract something from them. So this company, Pharma, a Spanish company, had developed trabectedin, which was used for ovarian cancer, and now lurbinectedin is sort of a synthetic analog of that. So very interesting premise for this company, and Jazz in-licensed this compound from this company. So let's talk about lurbinectedin. So it's a synthetic compound, meaning sort of related to trabectedin, as I mentioned, and it's an analog, and it's from the sea, and it comes from the sea squirt. I've developed a great affection for the sea squirt because this organism has really given us this drug, which has helped people quite a bit. Okay, so I'm going to take us back, and we're just going to focus on the left side here, just to give you a sense of how this drug works. Let's go back to high school biology, college biology, and as we all know, DNA is in all of our cells, and it specifies the instructions for how to build the building blocks for our body. It gives instructions for how to create enzymes, which help reactions and help things get built or processed in the body, right? And if things go normally, we have an enzyme which comes to the DNA and kind of reads the map, if you will, and then translates it into messenger RNA, which then goes out into the cell and is kind of produced in the factories, the instructions to how to make proteins and so forth. And that's good, that's normal, that's how it's supposed to go. The problem is in tumors like small cell lung cancer, you'll find that this process doesn't stop. So imagine this, you've got a road construction crew that's out here and they're trying to pave the road. They set up, they start the paving process, but they don't stop. They keep working all day, they work all night, they work 7 days a week, and they just can't stop. That's similar in a way to what's happening in small cell lung cancer. These tumors can't stop. So we call it oncogenic transcription. It just keeps going. And so something's got to put brakes on it. So what happens is a drug like lurbinectedin, it comes in, it binds the DNA and kind of blocks the enzymes from continuing that road construction, that paving, if you will. And then it takes some of the machinery and it kind of targets it for destruction. And then when the cancer cell tries to repair itself with damages in DNA, the drug interferes with that. So that's the crash course lesson in how this drug partly works. So that's one side of it, but there's actually another side to this, and we're gonna ignore the left side, and we're just gonna go to the right side here. In addition to blocking transcription of DNA, what it does is it goes to the tumor microenvironment. And the best way I can kind of say this, it's like being in a really bad neighborhood kind of thing, right? A little dangerous, a little hot, if you will. And that's what the tumor does. It creates a bad neighborhood where not such good things can happen. And if you have a drug or some other part of our immune system that can go in, kind of cool it down, get rid of some of the bad chemicals or growth factors and things that are being secreted, that can stand to help us a bit. And I'm going to use a Star Wars analogy for just a moment here. So see this purple thing here? So this is a tumor, what's called a tumor-associated macrophage. And so macrophages are parts of our immune system. They're helpful to us. They find and kill off invading microorganisms. So this is something that's good. But what kind of happens in a way with the macrophages, when they come in the presence of the tumor, it's, again, the Star Wars analogy, it's like they're converted to the dark side. They don't do what they're supposed to do. They, in fact, start doing the bidding of the tumor itself. So that becomes a real problem. And so one of the things that's hypothesized about this drug is that it goes in and kind of prevents the macrophages from coming to this area in the first place, or it takes the ones that have gone awry and destroys them. So again, a little bit of a crash course in how the drug works. Now, this drug was studied by Pharma in back a number of years ago in a phase 2 study, and they studied 9 different tumors, and the idea was to do what they call seeking a signal. They tried 9 tumors and said, where does it work? It's like taking the spaghetti and throwing it on the wall, and if it works somewhere, we will continue to pursue phase 3 and more studies. But what happened was, in the small cell lung cancer cohort actually had a great result. Like 35% of people responded, there was a good duration of response, and while there were still side effects, they were relatively benign to some other cancer drugs out there. So what this study did is it took about 105 people who, were functional, not the worst, but still had some impairments. They had one prior line of therapy before with platinum compound, and then about 105 patients, and then they looked at response rates and so forth. It was so good or so interesting that the FDA actually gave approval to this drug to be marketed. Now, this was an accelerated approval, which means they kind of give you a conditional approval, meaning the data looks good, we have a high unmet need with these patients, we got to get it out there, and that's good, we're going to give you the approval, but we want you to confirm this in what they call a confirmatory trial, like do it again, let's see if this actually replicates. And there is a trial going on by Pharma to see if this can get replicated. But in the meantime, while that trial's running through, again, this was a bit of a surprise that small cell lung cancer cohort did so well, but we have one problem. Usually you do a bunch of trials, you get to phase 3, you have a lot of evidence, so to speak, for how to the drug's effectiveness and safety and so forth. But here we had just this one trial. It was single-arm trial, meaning you gave a bunch of people the drug, but you didn't have a comparator. were they getting, in our case, we wouldn't give placebo, but did they get some other standard of care drug? There was no comparison. So we've spent a lot of time over the last few years trying to say, how can we build that evidence base to give people a sense, give them confidence in the drug, and how does it compare to other things? So there's sort of two categories here, real world studies. So we went and looked at population databases of patients who've received other treatments. And we kind of say, let's try to match up those patients with the patients we had in our trial and see what the performance looks like. And I'll be very sort of simple here about it, but when we did that, and this refers to the second one called external control arm, we're looking at how other people did who didn't take our drug, but coming from these databases. And in general, I would say, we did comparably. There was definitely efficacy. The safety was relatively benign, and there's a lot of papers and posters and things we can show you for anybody who's interested. But the other thing we did is we launched a phase 4 trial, which basically means for a drug that's already marketed, we just follow these patients. We say anybody that's getting Lurbinectedin, let's see how you do. What does your efficacy or effectiveness look like? What does your safety look like? And we've had some preliminary data come out as this study is maturing. And in general, I would say that the efficacy is roughly in line with the phase 2 data. It's a little worse because sometimes patients in the real world, have more comorbidities, other diseases, other complications going on. And the safety was relatively benign as well. So kind of confirming what we saw in the phase 2. And then I'll just point out this Lagoon trial being run by Pharma is the one that, if it's successful, would take that accelerated or conditional approval and convert it hopefully into a full approval. Okay, so I'm not going to go through all of this, but I'll just say with that phase 4 study I was talking about, we recruit patients who are taking Lurbinectedin, we treat them, collect a bunch of information on them, and then just follow them over time. And that trial is still continuing to recruit, and we're going to report out data as time goes on. Okay, so let me just quickly hit this trial that Pharma is doing. Again, this is the one that hopefully will convert the accelerated approval into a full approval. It's a phase 3. I'm not going to go through all the details, but suffice to say, there are three regimens being tested. Lurbinectedin by itself, Lurbinectedin with a drug called irinotecan, versus topotecan or irinotecan, which are considered sort of the comparator standards. And if successful, cross fingers, we'll have the full approval. But there's another thing that's going on that is a little bit different for us. So there's a phase 3 trial going on right now in partnership with Genentech, and it's looking at the use of Lurbinectedin in first line and in a very specific context. So as you all know, when you receive first line therapy, you're getting a platinum drug, you're getting a etoposide, and now you're also getting an immunotherapy like atezolizumab or Imfinzi. And the question is, well, what if in giving the atezolizumab to help patients, do well in the maintenance setting, we call it, after they sort of had a response. What if you add Lurbinectedin to that? Does it make it better? So that's what this trial is asking. If we take Lurbinectedin added to atezolizumab versus atezolizumab alone, and this is what's called in the maintenance setting, so a patient's had a response, and now you're trying to sustain that response. And so about 700 patients were enrolled, about 450 were expected to have a response, and then they're randomized or assigned to one of those two groups. And this trial is expected to read out in 2025, and if it's successful, what we would hope to see is that Lurbinectedin gets used in the second line where it is right now, second line and beyond, or possibly even first line in maintenance. So we'll see what happens. Okay, and I'm about to finish, but I just wanted to say, if you look at the Jazz website, there's a lot of patient resources there. So please, anyone feel free to avail yourself of that. And you can also send in a request if you've got specific questions or want any particular resources. We're happy to fulfill that. So I will stop there and thank you for your time.
Thank you so much. Badri, I call you Badri because I can't pronounce your last name. But also because I consider you a friend. Do we have any questions? Of course, Maida.
Sorry, you guys. I keep telling myself to shut up.
No, don't shut up.
This might be a question that no one can answer, but is there an average time, I mean months, years,
from the time a drug is first being tested in clinical till it's approved by the FDA for all of us?
Yeah, I think it's, I'm just speaking very generally here, and it'll differ by the sort of drug class, but I would say this process can take like 7 to 10 years and $1 billion or more to pull it all the way through. Now, of course, there are compounds along the way that don't succeed, but I think the latest stat is something roughly 1 in 10 that starts from the beginning gets to an approval.
Badri, thank you so much for you and for Jazz for your commitment to small cell lung cancer. This is very, very important. I want to comment on something you commented on and then maybe ask you a question to pose back to the group, which is you commented on this great response for Lurbi in the second line. And I'm going to maybe expand on your comment a little bit and then ask you to comment back to the group, because I think I'd like to expand a little bit on this great response, because I think it's important to put it in context.
Yeah.
And I agree it's a great response. So 35% response in a phase 2 trial, which is for small cell amazing. So before I comment further, can you give us and give the group the context for what is the typical response for small cell in the second line or higher? So why was 35% great? Because I think that's really staggering for the group to understand.
Yeah, I think if you look at like other drugs like topotecan, which have been used for a long time, depending on the studies you look at, I mean, you could have response rates like 16%, 20%, but you're, unfortunately not going to see like 80% or anything like that. So I think that's the context back a number of years. Now things have changed. You've got immunotherapies that have come on sort of in the 2018, 19, 20 period. So some things have changed. So I would say that would be like one of the markers or indicators when you look at some of the other therapies of the response you're getting. But I also want to say that you will find with a lot of therapies a pretty wide range of response depending on the studies that you're looking at.
Yeah, so can I make, just for the group, I really want to emphasize this point because I think it's a really important one, is for second line small cell and beyond. So when we're talking about after you've gone through platinum etoposide and then you're like, hey, what's next? You can actually say, hey, Badri, is 35%, meaning only 35% of patients had a response to the drug, and we call it great. Is that really great? Actually, yes, in small cell it is, because the historical context for second line therapy after platinum etoposide is like 5%. So some of the drugs that we've had for a long, long time, like something called topotecan, for example, response rates less than 10%. Immunotherapy in the second line, response rates of about 20%. And so on one side of the equation, I'm an eternal optimist. You have to be. Always, hey, we got to keep pushing. One side of the coin could be 35%. You're telling me only one of 3 patients responds. It's not good enough. We still have a lot of work to do. But compared to where we were, that is a big leap forward. And so we have to do better. That's why we're all here. But this is an advance. And I think to understand why it's an advance, you have to understand where we came from. And historically speaking, drugs for second-line small cell were miserable. And so putting it in context, I think, really emphasizes why Lurbi was such an advance in our world, why we're all excited about it. So just wanted to share that.
That's great. Thanks for that context. Very helpful.
Thank you, Dr. Lovely. And we also have an online question. We are being live streamed. So Christine asked, how long can someone remain on lurbinectedin?
If the patient is not having progression or switching off to another therapy for other reasons, you can just continue on. And I'll say, just very anecdotally, some patients, they go for a few cycles. Others have gone on in the teens of cycles. So it just depends on how the patient does.
Thank you. Questions? Okay. Wendy?
Can someone explain to me why immunotherapy is never offered at the limited stage?
I'm sorry, could you repeat the question?
Can you explain why immunotherapy is not offered if you have limited stage at your first line of treatment?
I think Dr. Lovely was about to answer. Did you want to?
So a very timely question. Just last week, there was a big press release for a trial called Adriatic, which is for Adriatic, like Adriatic Sea, which will absolutely change the standard of care for patients with limited stage small cell. And so drugs make their way into our standard of care treatments through clinical trials. And this clinical trial called Adriatic was introducing immunotherapy after concurrent chemotherapy and radiation. So typically what happens is there'll be a clinical trial with a new drug. Patients will enroll in that trial. So I can't send this message strongly enough. The only way we get new treatments for small cell is clinical trials, period. Period. So you guys, it's so important. I get it. We can't make new treatments unless we have clinical trials, and that I really like it. Anyone who's participating in a trial, thank you so much, because that is the only way we actually get drugs like immunotherapy into new settings, in this case, limited stage small cell. So what happens is there's a trial, the trial will enroll, the outcomes will come out. Typically there's a press release, and that's what happened last week. So the press release is like, immunotherapy improves overall survival for patients with limited stage small cell. And then the company will go to the regulatory authorities in the US, the FDA, and at some point it will come approved. So it's not yet approved, but it will be soon.